And hi everybody, welcome to the Spectral Flow Cytometry and Data Analysis Techniques webinar. This is a webinar that's going to be covering working with Sony Spectral Cytometer, data and instruments. We're joined on the line here by Greg Veltri from Sony Biotechnologies, and the team at Novo Software is very excited to be working with the team at Sony to providing a data analysis suite of tools in FCS Express that can work specifically with the spectral data, the raw spectral data that comes off of these instruments in an integrated format, so you can work with all the standard flow cytometry tools that you're used to working with in FCS Express directly with spectral plots. But today for the agenda, Greg is going to be giving us a presentation and an overview of the Sony Spectral technology and some of the software on his side. Then halfway through the webinar, we're going to switch over and start taking a look at some of the data analysis techniques directly within FCS Express with some of the same data. So at this point, I'm going to flip the presentation here over to Greg. I think you should have that.
Okay.
Great.
All right. Can you all set?
Looks good.
Okay, great. Hi, everyone. Sean, thank you for the invitation and this opportunity for us to talk about our collaboration that we've done together. For those out there, I'm sorry if I sound a little hoarse. I'm fighting a little bit of a cold, but I'll be more than willing to take questions later on, too. So what we're going to be talking about today is the instruments themselves. We'll just talk a little bit about their anatomy and design. And then we'll talk about some of the differences and advantages of Spectral. And then we have a feature that's very unique to Sony, which we've worked hard on, is standardization. And from there, we'll talk about a spectral library. And the spectral library allows us to keep all the fluorochrome single positive controls, and that's due to standardization. And then I'll go through an example of the acquisition and a little bit of the analysis, and then we'll go to Sean from there. So let's talk about the instruments themselves. So what I have here is the SP6800, and we also have the SA3800. Two differences are the SP has 3 lasers and the SA has 4 lasers. But what both of these have in common is how they collect the light. It's very unique to Sony. If we look closer, what you can see is that we collect all of the light with a 10 prism system. This 10 prism system has no bandpass filters, which is very unique, and we're the only ones that have that. And we collect all of the light from 420 nanometers to 800 nanometers. So now let's discuss a little bit about the differences and advantages of Spectral by us collecting just the pure light. So as we know, cells have fluorochromes on there, and they go through a laser and they emit the fluorescent emissions. Now, what's unique about this is that usually this fluorescent light overlaps, and this can be a problem. But in Spectral, we keep all of the light, and we sum the fluorescence together to do this by unmixing. Now, when we do this on conventional flow cytometry, we use color compensation to determine each fluorochrome intensity by subtracting overlapping fluorescence and the gaps. Thus, a lot of data that's important to us is lost. If we do a comparison, you can see that the spectral flow cytometry is collecting all of the light, and the conventional flow cytometry is only collecting a portion of that light. So let's look at some further differences and advantages. So autofluorescence tends to be an issue for a lot of people in flow cytometry. And what I have here is an example of GFP versus Alexa Fluor 700. Now the issue with that is that the common problem with cell autofluorescence is that it manages to masquerade as the true fluorescence. So if we look closer, what you can see is that what we think is a positive population is actually the autofluorescence. In spectral, what we're able to do is remove the autofluorescence and treat it as a separate color, so we're able to see the fluorescent population that we're interested in. So because of this, we've also developed a unique feature called standardization with our instruments. Now, standardization is talked about quite a bit in clinical flow and people who are doing long-term studies. So what we have here is a conventional flow cytometer. And what we know from conventional flow cytometers, and we're doing some form of standardization, is that we're trying to do a setup and a tracking. Now, what it does do, it measures the sensitivity, it tracks these parameters over time, and it chooses the gain setting for only one instrument. And that's key, that it's only one instrument. What it doesn't do, it doesn't take into account different autofluorescent populations, doesn't take into account the different lasers and filters of these systems. What standardization on a spectral does is, one, the standardization mode sets the system to master the settings to eliminate variability in a single instrument or among instruments that are located across multiple sites. Two, this unique capability allows experiments performed on any Sony cell analyzer to produce highly reliable, accurate, and reproducible results. And then third, this function also eliminates setup, subjectivity, or collaborative long-term studies with different operators. So if we look a little bit to see the standardization uses its automation and it completes more tasks more accurately, how does it do that? Well, let me show you. If we look at our instruments and spectral analyzers, what you'll see is that within 10 minutes, we're measuring the sensitivity. We are choosing the gain settings, the laser delays, optic checks, and laser alignment, where our competitor is only doing 3 of these multiple things that we're doing on our instrument. Plus, we're taking it a step further. So our standardization is automated, we're giving you more QC, and it's a more accurate standardized form. Now, because of standardization, we're able to have a unique feature called the Spectral Library. So the first question is, what is the Spectral Library? Well, the Spectral Library is a personal reagent library that simplifies experiment creation and saves time. So if we look at this library, what we'll see is that it's done by acquiring the controls, and it's automatically stored into the library as you do it. The information that we can place into this library is, 1, is the fluorochrome, of course. 2, the antibody and their clone. 3, we can collect on a bead or a cell. And then 4, the reagent's expiration date. A unique feature, though, is that as we collect this, we can get the spectral brightness index. And this is based on the difference of the negative curve and the positive curve. So it's similar to a staining index, but a little bit different. So this is a simple 3-step process. The first step is that you acquire your control. This can be on a capture bead, or it could be on cells. It's up to you. You have to have a single positive control. And part of that you also have, it's unstained. This information automatically goes into the Spectral Library. Now, because of standardization, you can share this library with other people or with other labs. The second step now is you run your samples. All you have to do with your samples is one, bring one that has everything stained, and then two, you're negative, so that we know what the autofluorescence is of the population that we're looking at. The third step then allows us to then analyze your data either at the instrument or at a workstation. So let's go through a little bit of an example of the acquisition of placing something into the Spectral Library. So what we have here is the software of the SA3800, and the SP6800 has the same feature. Excuse me. And what we're doing here is we're just placing in some of the fluorochromes that we're going to be using for this experiment. And what you'll see is that we can place in the marker, the species, the clone, etc. We can also place in the manufacturer, the lot number. We can get very specific, or we can keep it simple, and the only thing that we have to do is say, what is our laser configuration for this instrument? Once we do that, we can then do this on the SA, either a 24 tube rack, a 96 well, or 384. We can tell it that we have a unstained control, or we can tell it that we're doing it without an unstained control. The acquisition setting allows you to have agitation on this, and allows you to collect it in different ways. The worksheet allows you to define your single positive controls by a spectral plot or a histogram plot. You can then place in different information into this, and then you can then do a preview and see where you are. If you accept this, you can then now go and use a wizard or no wizard. The wizard just gives you a step-by-step of how to use the instrument and save the single positive controls. It tells you how to set up your flow rate, your lasers, your detectors and threshold, and it goes through each one step by step. If you feel like you're an advanced user, you can just use a template and just place this information into your Spectral Library. So what you're seeing right now is that you're able to see what's being collected, and if it has a check mark, what has been collected. The arrow that you see here means this is where it's starting and going on to the next one. We also have information about our event rate, telling you how fast it's collecting and how slow it's collecting. But once you acquire, the next step is just to say what's positive and what's negative. So in this example, we did this by spectral plots. And what you'll see here in the spectral plot is this bead has the positive curve, and this is the negative curve. If you're not sure, it's not a problem. These gates are automatically placed there for you, so that all you have to do is make little adjustments and not have to worry exactly to place it in the exact position. Excuse me. Now once you define these, which we're doing right now by placing a gate around our bead population in the forward scatter, side scatter, you'll see that all of these have been defined to what's negative and what's positive. Excuse me. And you can see what I'm doing here is I'm adjusting it to however I want it to be. So you have a lot of flexibility with the software. But the next step is then to say calculate. All the calculation is doing is it's looking at the curve and defining what's negative and what's positive. The next step is then to just place it into the library. So you'll see that there's an add to the library. You can put additional information if you like to, and then click add to the library. And it's that simple. Now once this information is in the library, you're all set. You don't have to worry about single positive controls. If you worry about tandems falling apart, that's what the expiration date's there for, and it will give you a little warning saying this particular fluorochrome is expired. Would you like to replace it, or would you like to continue? So let's go through an example of a sample that's been acquired and using the Spectral Library. So what I have here is a 10 color sample on the SA3800. And I have two tubes. This tube is obviously the sample tube, and this tube is the unstained. Okay, this is human peripheral blood. It's been lice washed. And what we're going to do is we're going to start with the sample. Now, what we have here is the color palette. The color palette has all of the markers that we're working with. It has all of the fluorochromes. But what we don't have is what's in the unmixing. The unmixing allows us to separate each of the spectral fluorochromes so that we can see the populations on our dot plots. So what I would do if I was a user at any lab or any core lab, I'd go load my spectral reference, which comes from the library. It automatically knows which fluorochromes have been placed into my color palette. So all I need to do is shift and select all. And now I have all of these with all their markers. What's also nice about this is that I can see each of the unique curves that have been collected, and I can see if there's anything different or unique about these. Once I feel comfortable with everything, I select load. And what you'll see is right down here in this lower portion of the window are all the curves that I've placed into here. These curves are the reference curves to tell the algorithm how to separate each of the populations. Now, we mentioned before that autofluorescence can be an issue for people, and that autofluorescence can hide or masquerade as a false positive. So we're going to go to the unstained, and what I'm going to do, I know from previous experiments that actually the autofluorescence for my lymphocytes, monocytes, and granulocytes are basically very similar to each other. They have the same spectral curve to them. So what I'm going to do is just define that, and then I'm going to tell it to calculate. Now what you're going to see at the end of this calculation is that there will be a purple population sitting within the curve. This purple population is the curve or the color of autofluorescence. It has a unique shape to it based on the type of cells, and that's due to any oxidation items, the way you fix it, the way you may permeabilize it, and the NADH and the FADH of that particular cell. So though we continue to complain about autofluorescence, it actually has a lot of power to it. It could also tell you drug loading and other items about the cell. And this is something we have on our webpage that has technical notes about using autofluorescence as a unique color. So once it's done calculating, My computer's a little slow, my apologies. We'll be able to see the autofluorescence population. Now, what you'll also notice is that here on the negative of the unmixing, there is no population. And the reason being is that when we take it from the Spectral Library, which is the SRL that you see here, that negative is automatically collected from our previous collection, and it's just looking at what's the positive curve. So here's the autofluorescence population in that purple color that I was talking about. And you can see where it sits. We have autofluorescence sitting in the PAC blue area, in this, excuse me, FTSE area, PE, and it continues all the way down. So let's go back to our sample now that we've done that. Okay, and we'll hit apply. And what you'll see is that the populations have uniquely separated into different ones. Now, those who've done flow for a while know that we have a little bit of a spread. And now spread can be from two different things. In this case, it can either be from an autofluorescent contamination, or it can be just the way I stain that sample. Okay, so at Sony, we recognize that there's a lot of unique features to Spectral and how we collect our data, how we stain our data. And because of that, we have an item called the Spectral Reference Adjuster. And what that allows us to do is to adjust the curve of the spectral so that we can decrease spread. So we're looking at the lymphocytes. So let's select the lymphocytes. And what we're going to see on this is all the different populations, possibilities. Okay. And let's increase this a little bit to 50,000 events so that we can see it a little bit better. All right. Now, if we look at this as flow cytometrists, we know that populations going all the way down is kind of an overcompensation. In this case, it's overmixed. So you'll notice that as I place my arrow on this population here, you'll see that the BV421 is lighting up on the spectral. So I'm just going to pull this up a little bit. And all I've done is adjusted the curve just a little bit in the BV421. Okay. And what we can do is we can go through here and look at the populations, and you can see you can place it down or you can place it too low. You can see the populations bending over a little bit. And you can see you could do it too much. So you have to be a little careful, of course, and it's just like anything, what we do in science, is that it has to make sense also at the same time. And then we can scroll down and look for any other ones. And if you remember from the previous plot, this population that was the one that was kind of sticking out for us to see that it was spreading out too much and going too low. So we're going to pull this up a bit. And you can see it adjusting the curve right over here. And I'll do it some more. Okay. Once we feel good about that, we click OK. And we wait for it, and then we apply it. And you can see now that this population that was spread was sitting quite large, and it was sitting lower than what we expected, is now sitting in the median area where we expected this to be. And if we close this, we can see the plots a little bit better. And you can see this decreased the spread on my basophils, and etc. Now these are just the things that we do to kind of prep it up, so now we can send it to FCS Express. And what we'll do is we'll select this right here, and we'll do export to FCS files. So we have FCS file only, and we have FCS file and SRAW file. So we're going to select that. We're going to use unmixing adjusted because we did the adjustment in the spectral reference. Now, if you wanted the data without it being unmixed, you can get that also, a pre-unmixed data. And if for some reason you wanted to look at it in a matrix of conventional, we can go back and we can look at it conventionally also. And then what I would do then is just export that. And then those files would then go to wherever I've designed it to go. In this case, I've asked it to go to the desktop. And I'm just going to stop right there. Okay. So now that we've exported these files, Sean, we can now have you look at it.
Great. Thanks a lot, Greg. So what we'll do here is bring up my screen again. And again, if you have any questions about the kind of technology and acquisition portion of the webinar, I do encourage you to enter them into the question and answers panel at this point. We'll make sure to come back at the end to kind of address those questions as well. And what we'll do now is start having a look at what you can do with that data in FCS Express. So as you just saw, Greg had chosen to export the FCS files and what's called SRAW file types. And what that will do is allow you to load up not just the FCS files, but the full spectral data within FCS Express. And kind of working, you'll have the ability to kind of work between standard FCS files and the spectral data. So again, what we're going to be covering today is working with those Sony spectral data files and the FCS files. And we're going to be talking about working with them in a special plot type that we have in FCS Express called a spectrum plot. And that's the plot type that's going to allow you to access that raw spectral data as it appeared in the Sony instruments and software. So again, these things are linked up together, and this allows you to use all the functionality that's been developed over the last almost 20 years in FCS Express. So that means you can take this data, you can analyze it on a Mac or PC, you can do that interchangeably, and you can really do it from any computer in the world. You get to analyze in a Microsoft PowerPoint-like interface in FCS Express, and there's a lot of kind of unique and intuitive drag and drop capabilities in the software, as well as real-time gating statistics. And that means that if you move a gate, even just 1 pixel, any sort of kind of statistic or backgated populations, including those spectral plots, will update for you in real time in FCS Express. We also have tools in the software for integrating things like spreadsheets. If you've ever kind of wished that you could link up Microsoft Excel to your flow or spectral cytometry analysis, essentially we can do that now in FCS Express, as well as tools for plate-based analysis and high-dimensional data reduction tools. So we do know that, when you're acquiring with these Sony instruments, one of the really great benefits is you can acquire lots and lots of parameters without having to worry about the traditional kind of compensation and spillover that may limit you with, traditional cytometry systems. So with that, there's also kind of a need to distill that data down with tools like Tisney and Spade. So we'll kind of go through all of these things today. But to get started, let's talk about how we get that data into FCS Express and then how we work with spectral plots. So to start, you'll notice that what I have up here now looks very much like Microsoft Office or Microsoft PowerPoint. And that is shamelessly by design. We're actually in FCS Express right now. And if I create a new, what we call a layout, it will open up and there'll be a blank page on this layout that looks just like PowerPoint. And if you were working in PowerPoint, you'd probably be going to the Insert tab to create a PowerPoint presentation and choosing to insert something like shapes, text boxes, pictures, that sort of thing. But you'll notice that in our Insert tab in FCS Express, we have options to insert things like histograms, these spectrum plots, which I alluded to, as well as all of the standard flow cytometry plots that you could be used to using. So again, if you were working in Microsoft PowerPoint, you would probably start by clicking on the text box tab. You would draw a text box and you would put some text in there. And you can see that we have a text box in FCS Express. We can move it, we can resize it. Again, very much like Microsoft PowerPoint. Now, what we want to do is not just work with text boxes. We do want to pull our data into here and start looking at it. So what I'm going to do is show you the data that Greg had exported here. You'll see that we have the D03 well, which was the unstained control, as well as the D04 well, which was the stained sample. And we have the FCS file, as well as the SRAW file type. Now again, you can work with just FCS files in FCS Express if you want, but if you want to have access to the spectral data in real time in FCS Express, you'll need to export both the FCS files and the SRAW. Now there's a number of ways to get data into the software, and one kind of very convenient way, which you may do in PowerPoint already, is through drag and drop. And we can do that. If you have the data files somewhere on your desktop, you can simply drag and drop the FCS file into the layout in FCS Express, and you'll be prompted to choose a plot type. So in this instance, we'll choose a density plot, and you'll see that our data is now here. Right? We have a density plot. If you click and hold on the axes, you notice that we can change and pick and choose between any of the different parameters that we've acquired with this data set. And lo and behold, here's our monocytes, granulocytes, and lymphocytes populations that we saw in the Sony acquisition software. So again, this looks and feels like PowerPoint. Keep this in mind as you're kind of working in the software. Now again, there's lots of different plot types we can work from. So if we want to choose something like a color dot plot, I'll insert this and we'll kind of see what this does in just a moment. But really one of the reasons we're here today is to talk about spectrum, talk about spectral data. So we can insert a spectrum plot as well. I will choose that and I will draw the spectrum plot. And we're going to be prompted to go get that same data, but load it up with the SRAW file. So I'm going to choose D04, click to open the file. And what you're going to see is that we now have a spectral plot just like you'd be used to looking at within the Sony software. In this case, I'm going to change the area wavelength. And you can see that we have a plot that's very similar to what was being shown within the acquisition software from the Sony side. So again, once you have these plots created, it's very straightforward in FCS Express to start setting up gates and start creating an analysis just like you would be doing a PowerPoint presentation. So again, you would be clicking on something like the gating tab, drawing a gate around a population. You'd be naming that gate. In this case, we'll call it granulocytes, and you can see that any other plots that we have inserted will automatically show the color of that gate on the plot. This is what we refer to backgating in flow cytometry, and we're going to be coming back to that a few times today as it relates to spectral plots. But you can apply that gate, right? So if we apply that gate through dragging and dropping, you can see how this information updates in real time. And if we change to any of the different parameters, you can see again, as we move these gates, it updates in real time. Now this also goes for spectral data. So if we have a spectral plot and we have a gate created, we can drag that gate onto the spectral plot and we can see in real time as we adjust this gate what the spectra for each of these events that fall within that gate look like, right? So you can see how fast this is actually working. We've put a lot of effort into making FCS Express update in real time, especially when you're working with these large spectral data files, so you get real-time information. Now again, you can create a gate directly on the spectral plots as well. So if we needed to come in and investigate what's going on at a particular position along the curve, we can do so. And what you're going to see, once I get these gates out of the hierarchy, is that as I adjust this gate, it's going to update that information in real time in any of the plots that are there. So again, this is some of the basics of working with kind of plots and inserting these plots in FCS Express. And the one thing that you need to keep in mind is that we can always export directly to PowerPoint into Excel, and whatever you set up here is essentially a final report that can be standardized and used over and over again. So what I'm going to bring up essentially is a mock-up of what was done in the Sony acquisition software within FCS Express. And again, you can see here that this looks and feels just like Microsoft PowerPoint objects. We've set up all the kind of gates and particular analysis routines for this report in FCS Express that we had set up within the Sony software. And we get real-time updating. So again, if we adjust our lymphocytes gate, we're going to see on the spectral plot on the bottom right, we're going to see all of our plots updating here in real time. And this is really great to use as a template for analysis. So if we have two files, in this case we have an unstained and a stained, you may have hundreds of wells that you're looking at or hundreds of tubes, you can simply load them into what we call the data list. And you can click on the iteration number and you'll see that we will navigate between these different data files to quickly and easily update what's happening in that report, both for the spectral plots and for the standard flow cytometry plots. And this really comes into play when we're getting ready to kind of analyze lots of data and you need to export this. You can simply come up to the Export tab and export this directly to PowerPoint, to PDF, wherever it is that you really want to go. So I'm going to show you kind of what this looks like in Microsoft PowerPoint, because again, what we're doing here is not just an analysis. What we're doing in FCS Express is really your final result. So that when you've loaded your data into these analysis templates, you've set your gates, you can export directly to PowerPoint. You can see here that we have all the information from all the pages. We'll kind of go through what each of these involve in just a few minutes, but they're all super high resolution graphics that you can break out individually from each other, and you're ready for reporting or publication with that data. Now, I should mention, you see on the second page, there's actually a pie chart involved here. So kind of getting back to what I mentioned, that in FCS Express, your analysis is your final result. You can insert Excel-like spreadsheets. You can insert pie charts and bar charts and regression lines, and these all will link up to your data. So as I adjust this granulocytes gate, you can see that my pie chart and my Excel spreadsheet like values here are updating for me in real time. And the way that we handle and create these chart types is through our Insert tab, and we would insert a spreadsheet. And you can see here that this spreadsheet that we've created, when we click on it, we have full formatting and tools that you would have available within Microsoft Excel, including any kind of formulas, financials, math and trigonometry, statistical informations, if you need to do t-tests and p-values and things like that. Really everything you can do is from Excel is included here. And these spreadsheets are very easy to populate. You can either manually enter in values, or better yet, if you wanted to drag this whole granulocytes gate in and say put in the number of events, we can kind of repeat this with whatever kind of gated populations or statistics we want in the software. And you can see that we can build up this information very quickly into that spreadsheet, and again, it will update for us in real time. And creating the charts, again, very simple. We simply come in here and we say insert something like a bar chart, and then we get our results for whatever it is that we want to look at within that Excel spreadsheet very quickly and easily. So these are some tools to keep in mind as you're kind of progressing through working with your data, that when you're working in FCS Express, your final result is in this analysis. You don't have to copy and paste out to things like PowerPoint or Photoshop or other kind of software packages to help you get to that final result. But again, what we're here for today is to talk about specifically some of the spectral plots and spectral features within FCS Express. So when you insert a spectral plot in FCS Express, by default, you're going to get this type of view. Essentially, it's a density plot type of view, outlining kind of where the density of all the events as they appear in all of the spectra fall within there. And with the spectral plots, you can simply format these and change them to get whatever type of display type is most useful for you to analyze this data. And we can do this with any plot. Essentially, we right-click and choose Format, or double-click on the plot, and you'll be presented with all of the options for these plots, right? So if you needed to come in and change scaling, change what's on the axes, what's in the titles, what's in the legends, you can do so. And when you're working with spectral plots, there's a series of special options for working with spectrum plot specific options, working with things like color levels, which gates to display for backgating. And I'll go through these somewhat one by one to show you how these work for the spectral data that you have. So at a very basic level, one of the easiest things to change is essentially the color levels. So in this density plot, our default is to display what we call a flame. If we wanted to change that kind of flame coloring scheme to something called rainbow, we can do that. If we wanted to change this to a grayscale, maybe avoid some kind of color page charges when we're publishing, we can do so. But again, there's a number of kind of different color schemes that you could be using here to change how that data and that information is presented to you. Now, getting a little bit deeper into some of the spectrum-specific options are how we kind of smooth and display this data within FCS Express. So by default, we use a somewhat lower resolution for these plots, but this resolution can be changed at any time. If we were to up this resolution from 100 to 200, you'll notice that in the density plot on the right, things have been smoothed a bit. And depending on how you set that resolution, the smoothing can be increased or decreased. And we also have an option here to interpolate values, or essentially connect these different data points so we can draw proper lines from them. So if we choose to interpolate the values, what you'll see here is that we're actually starting to connect the points, getting kind of more well-defined lines. We're getting some more smoothing from this data. So that way we can really generate a very nice looking plot that's going to help us define very quickly and easily by looking at it, what this spectra is telling us, what's involved here, and eventually how we can create gates on this. Now, there's a number of options here as well. Instead of just displaying with a color on density, we have an option here called color on gated overlays. And essentially, this is going to give you backgating information for all of your spectral lines that are within that plot. So if I come down to this plot, you'll notice that we have a number of spectral lines that are colored differently. And what you'll see is when we format this, it's because in our spectrum plot specific options, we've chose to color on gated overlays, and we've also included something called shade based on density. Now, the shade based on density mode will give us a kind of color shading of the density of the events that fall within these lines. So if I uncheck that, you'll see that line appearing and disappearing. And again, what this color on gated overlays means is that any of the events that are blue here are coming from something like our B cells gate. Any of the events that are kind of violet here are coming from our CD45, CD3 negative gate. And this can all be adjusted through our color or through our display gates options. So if you did not want to view the backgated information from all the gated populations at one time, which can become overwhelming, especially if you're working with 10 color data, you can come in here and you can say, well, just pick and choose which gates you want to display. And in this case, I will show the lymphocytes, the monocytes, and the granulocytes. And when I preview that option, you'll see that I have backgating now in red for our lymphocytes. We have the monocytes in this kind of bluish color. And as I adjust this gate, that backgating information is going to update for us in real time across any of the spectral plots and any of the other type of plots that we can show backgating information on within FCS Express. That goes for over here as well. right? We have another plot that's set up, and if we were to set up an additional gate on the spectral plot, what we're going to see when we make this gate bright green is that that population essentially is going to be backgated, so we can really kind of see, how these curves and these spectral lines are represented for any of the populations that fall on any of the other plots. So some very kind of convenient tools to work with these things. Now, we also have some other options in here as well for these plot types. Other than just the spectral plot-specific options, we have options for the overlays in particular. So if you wanted to show the average instead of showing the entire, all of the spectral lines, we can do so, and you'll see that now we have an average of all those events rather than just showing every individual spectral line. And this becomes very important if you're working with things like overlays, which we've done here on this page. What you'll see is that instead of showing every single spectrum, in this case we're very interested in looking at the spectral of the lymphocytes versus the B cells and looking at the average of these two gated populations. So that way, as I adjust one gate or as I adjust the other gate, I get that information in real time, and I can really kind of come in here and decide if I need to set some additional gates on my spectral data and how that can be useful. Now, we've also done this in the bottom here with CD45 positive, CD3 positive, and negative populations, and also included a negative overlay here. So I'm going to show you very quickly how do you actually create these things, how do you create an overlay. So the first thing I'm going to do is actually remove my negative overlay. And to create an overlay on any of these spectral plots, you right-click on the plot and choose to add an overlay. I prefer using the Advanced Open Data dialog, and what this will allow me to do is choose an active file from the layout. So in this case, I'm going to choose DO4. I'm going to tell FCS Express which spectrum parameter I want. So it's going to be the area wavelength parameter. And I'm going to say that for this overlay, let's use the negative gate and click OK. And when we add an overlay, by default, all of the spectrum are going to be shown. So we'll have to do one more quick formatting step here to come in and say, just show us the average for the negative population. So in that way, with just a few clicks, we've essentially created an overlay of the different spectrums for 3 different populations from this graph. And as we investigate these things by moving them around, we'll have access to that information in real time. So again, we've kind of gone through a number of the kind of functionalities, basic functionalities for working with spectral plots and working with this data in FCS Express, going from overlays, going through some of the different display options for working with spectral plots, as well as working with some of the reporting tools in spreadsheets and charts, and how to create some of these kind of general reports in the software. But there's also some additional features within FCS Express for plate-based analysis and high-dimensional data reduction tools. So I'm going to spend just a moment talking about loading essentially your FCS files without the SRAW data files into FCS Express. It's very quite straightforward here. And if we want to view a plate's worth of data at any time, essentially we'll come up to our Insert tab here and insert what we call a heat map. So if you do have the high content functionality enabled within FCS Express, what we'll be able to do is choose an FCS plate folder. We'll be able to click on one of the files that exist within that plate folder and click Open. And we will populate that plate with however many FCS files were acquired for that plate. So in this case, it wasn't an entire 96 well plate, so we could even zoom in on this. And the really great thing about working with heat maps in FCS Express is that if I insert any other type of chart, maybe we'll look at side scatter versus forward scatter, and I'll put this in 2 different places, we can actually start creating gates on that heat map. So if I know that I have 4 replicates in column 1, I can call this gate column 1. And if I know I have another series of 4 replicates in column 2, I can create another gate and call it column 2. And what I'll be able to do within my plots is assign the column 1 gate and column 2 gate and compare any of this data side by side. And if you need to edit these gates or change these gates in any way on the heat maps, you can do so and see how that information is updating and changing for you in real time. So really, there's a lot that we can do with plate maps, more than we can explain in 20 or 30 minutes in a webinar, or even just a few minutes. But we do have a full suite of resources about working with plate-based analysis in FCS Express. Now, another thing that you'll be wanting to do, or you may be interested in doing, is working with high-dimensional data reduction tools within FCS Express. So when you're working with data sets like this, even if you're working with 10 different colors, or let me undo a few changes here, get us back to where we were. So it doesn't matter if you're working with kind of 10 colors or 2 colors or 20 colors. If you need to, kind of distill that data down a little bit and start exploring it because it is very high dimensional, we can do this with our transformation tools in FCS Express. This is something that comes in every version of FCS Express. You don't have to pay extra for it. And you would simply set up a t-SNE transformation, and you would pick and choose all the fluorescent parameters that you want to participate. So the user interface for working with t-SNE and tools like SPADE in FCS Express is very straightforward. We have a lot of materials to explain kind of how we work with these algorithms and what they actually mean. But what I'm going to do is simply set up a t-SNE calculation with a sample size of 2,000. And I'm going to generate a plot to visualize that data. So I will insert a density plot. And what I will do to calculate and apply that transformation is simply drag it and drop it to that density plot. So when the plot title says transformed, that's how long our t-SNE transformation took, or our Barnes-Hutt approximation of the t-SNE transformation took to apply and calculate on that data. And it is very quick, it is very straightforward for working with this in FCS Express. We can choose t-SNE Y versus t-SNE X. And then we'll also come in here and make some formatting changes to this plot so it becomes a little bit easier to kind of visualize how this data looks. So essentially we'll do some smoothing and put a legend on this. And what we'll be able to do at this point is, from the legend, pick and choose which parameters we want to view the heat of on this t-SNE mapping. And what you'll see here is when we look at something like CD19, we have some kind of dim populations, we have some bright populations for CD19, and this is a great kind of exploratory tool to kind of see where these things are. Now, the really nice thing about working with transformations in FCS Express is that they can be applied to any plot. So if I'd like to apply this to our color dot plot and also apply this to our spectral plot, we can. And what that means is that these are now t-SNE mapped, and if I create a gate on what appears to be a very high CD19 population, and we'll make this bright green, it will be shown over here on our CD19 versus 4 plot. You can see that this is the CD19 positive population that was found within the t-SNE plot. And if I wanted to apply this back to my spectral data, you can see that we'll get the information, the spectral information associated with this population of interest that we found within t-SNE mapping. And as we adjust this, all of these spectrum for all of these t-SNE mapped kind of populations and continents here are going to be updating for us in real time. So this is a very, very powerful tool for data exploration, especially when it comes to kind of highly multi-parametric and highly multi-dimensional data sets. Again, these are things that are all included, and some of these features we've covered in just about 5 or 10 minutes each, but we do have full resources on the Novo software website to kind of help you better understand how and why these things work. So to summarize kind of some of the things that we talked about today from spectral flow cytometry data analysis and FCS Express is, how easy it is to use FCS Express since it works and looks and feels like PowerPoint, and how we have real-time updating in the software so that way you can have quick and efficient data analysis and explore populations either at the spectral level or at the standard kind of FCS file level. These spectral plots can be accessed and gated at any point during an analysis, so you can create gates directly on them. You can apply gates from a spectral plot to another plot, from another plot to a spectral plot, and even from any of our transformation tools directly back to plots. And as you saw, we have additional tools in the software for plate-based analysis of the standard FCS files and high-dimensional data reduction tools for both the spectral and the standard FCS files. So if you haven't yet had a chance to work with FCS Express, you can go to the Novo software website and you can download a free trial version of the software, or if you're lucky enough to have recently purchased a Sony instrument, FCS Express is going to be distributed or has been distributed with many of those, and you can download that demo just by clicking here. Again, you can use that free for 30 days to kind of try things out. And if you have any questions after the webinar about FCS Express, we really encourage you to contact support@denovosoftware.com. So with that, we also have some of the contact information here for Sony. So if you do need to be in touch with Sony or you have any questions about the instrumentation, acquisition software, and things like that, you can contact Greg Veltri at sony.com. You can visit the Sony website and use some of the information that's up here to help you at any time. So with that, I think we're going to open up the floor to questions, and thanks again everybody for joining us today for the webinar. We really do appreciate taking the time to come out and kind of learn a little bit more about this new technology, and I especially like to thank Greg Veltri and the team at Sony for putting this together. Every time I personally hear more about the spectral technology, I learn a little something myself. So thanks, Greg, and thanks everybody for joining us today.